Good afternoon, and thanks for joining today's webinar, Updates on COVID-19 Therapeutics, Vaccines, and Guidance Documents. I'm Oren Fix, and I'm pleased to be moderating today's webinar alongside my co-moderator, Emily Blumberg. Today's webinar is being recorded and will be made available on the AASLD COVID-19 Resources webpage. In addition to this webinar, the COVID-19 Clinical Oversight and Education Subcommittee has just updated our expert panel consensus document on vaccines to prevent COVID-19 in patients with liver disease, and that's now available for download on the COVID-19 resources page on the AASLD website. And you can access that page and any downloads there at aasld.org/COVID-19. This webinar is being brought to you by AASLD and its over 6,000 members. Our members are instrumental in creating exceptional educational content, such as this webinar, and I invite you to join us. You can play a vital role in bringing liver disease and treatment to the forefront and take advantage of the many member benefits we offer. Please visit aasld.org to learn more about becoming a member. Today's agenda includes some housekeeping items, presenter introductions, presentations by our distinguished speakers, and panel discussion followed by Q&A. You're encouraged to submit questions throughout today's presentation using the Q&A box at the top or bottom of your screen. We'll answer as many questions submitted to the Q&A live as time permits after the formal presentation. We will not be taking any verbal questions during today's webinar, but we will be able to answer questions by typing to the question presenter through the Q&A box. So feel free to start asking questions at any point. So now moving on to our introductions. One of our moderators, my co-moderator, is Emily Blumberg. She's director of the Transplant Infectious Diseases Program and the Infectious Diseases Fellowship at the University of Pennsylvania. And I'm Oren Fix. I'm currently affiliated with Washington State University, Elsa S. Floyd College of Medicine, but between jobs, as you can plainly tell. And I'm co-chair of the AASLD Clinical Oversight and Education Subcommittee, along with Bob Fontana, one of our panelists. Our presenters today are Adam Lauring, who's assistant professor in the departments of internal medicine at the University of Michigan, Daniel Kaul, professor of internal medicine, also at the University of Michigan, and a contributor to our expert panel consensus documents. Onyema Ogbuagu, who's associate professor of medicine and director of the HIV clinical trials program of the Yale AIDS program, and also in the section of infectious diseases at the Yale School of Medicine. And Mark Russo, who's medical director of liver transplantation and chief of the division of hepatology at Atrium Carolinas Medical Center, also a longtime contributor to our expert panel consensus documents and webinars. And with that, I'd like to introduce Dr. Kaul, who will be presenting the first talk. Dr. Kaul.
Thanks so much, Oren. I appreciate the introduction, and it's a real pleasure to be here. So I'm going to start and talk about monoclonal antibody treatments for COVID-19. They're my, you can go ahead to the next slide, actually. So those are the disclosure. And I just want to mention, as is common now with COVID, some of this data really hasn't been peer reviewed, and really some of it is almost kind of at the press release level, but sometimes that's a little bit what we're stuck with. Next slide. So the topics I'll cover is the role of monoclonal antibody in therapy. I'm going to talk a little bit about the need really for early identification of people who may benefit, because the data pretty clearly demonstrates that if this is going to work, if monoclonal antibody is going to work, for the majority of folks, you really got to get it into them early. Some of the challenges of administration, although there is work on subcutaneous preparations that may ease some of that, for now these are intravenous and require some kind of an infusion setting. I'll just briefly mention the ability of some of the variants potentially to escape from these antibodies, although I think Dr. Lauring will expand on that. And just briefly again on what, if any, do we know about the role of these in heavily immunosuppressed patients with persistent infection. Next slide, please. And so, sorry, this is a little hard to read. I'd modified it, but wasn't able to get it in time. But basically, the reason I want to focus on monoclonal antibody is that's really what we have available outside of supportive care in the ambulatory setting. And I think that's really important, particularly for a group of people focused on liver disease, because hopefully you'll have the assistance of whether it's infectious disease, hospital medicine, pulmonary critical care for some of the other treatments, remdesivir and corticosteroids and so forth in hospitalized people with liver disease. But it's up to all of us to try and identify people and have a system for those who might benefit from this monoclonal antibody therapy early on. Next slide, please. And so in general, these monoclonal antibodies are engineered to the receptor binding domain of the spike protein, which you can see here on the SARS-CoV-2 molecule. In general, this graphic is the Lilly product, but the idea is to use more than one monoclonal antibody so that the number of mutations or the impact of those mutations has to be greater in order to entirely escape these products, since they probably don't create as widespread immunity as a vaccine or potentially natural infection might. Next slide, please. So right now there are two preparations, two manufacturers that have expanded use authorization from the FDA. One of them is the Regeneron product, which is two monoclonals together. That's the one that the former president received actually when he was hospitalized in a situation where at least the current data suggests that it may not be effective. I don't think that was understood necessarily at the time. The Lilly product, there are actually two EUAs. So there's bamlanivimab, and then there's bamlanivimab plus etesevimab. It's a little hard to say. I think I may just say BAM from here and as a combination product. And really all the development is going towards combination products. I don't think Lilly has intentions to move too much forward with BAM alone, maybe as a preventative, but certainly not as a treatment for people with COVID. Next slide, please. And so what we've learned so far in multiple trials, some of them industry sponsored, this ACTIV-3, which is kind of an NIH industry consortium, have demonstrated that when you use these products, at least on aggregate, so this may not be entirely true of people who are heavily immunosuppressed, but otherwise they don't really work in hospitalized patients. And these trials were generally halted when they reached DSMB measures of futility. So whether that's ACTIV-3, which was the Lilly product, or the Regeneron product, there's still a little bit of activity in this area, but generally they may not be very useful for people by the time they're sick enough to be hospitalized. Next slide, please. And this Regeneron publication explains, I think, a little bit of why that's the case. So in this trial, there were two different doses, the 2.4, the 8-gram dose of this Regeneron dual cocktail, and it was given pretty early. So they got it to people 3 days median of symptoms. To enroll, it had to be 7 days, but they did an excellent job actually getting it from 3 days of symptom onset. And the primary endpoints were virologic. And if you look at the panel as you face the screen, the lower left are patients who are antibody negative at enrollment. And you can see there was a significant reduction in antibody levels. You couldn't see that in people who are antibody positive and presumably had been either infected a little bit longer or generated their own antibody and weren't going to get that much benefit from additional antibody. And it was interesting to me that actually most of the patients were actually antibody positive at baseline, even though they'd enrolled fairly early. There was a little bit of clinical data, so a medically attended event, whether that was going to a primary care, emergency department, hospitalization, small numbers, about a 50% reduction. Next slide, please. Now, the Lilly experience is really hard to put together from what primary publications there are, because they're up to like arm L in the BLAZE program, which is what they call most of their trials on this for treatment. But if you put it all together, so Lilly put it all together, so these are publicly available slides that Lilly put together. They're not from an academic publication, but you see a number of things. And when you look at going to be hospitalized or an emergency room visit, initially they'd included sort of other medically attended visits, but if you focus on hospitalization or emergency room, what you see is that in the placebo group, you have an event rate that approaches 6%. and much lower, whether it's the monoclonal at different doses, the combination. But when you do get to, or both of them combined, when you do get to the combination, you actually only had one medically attended event versus 9 in placebo, although obviously a larger number in placebo. When you look at people at higher risk, so that's the panel on your right as you look at the screen, age greater than 65, BMI greater than 35, there, with the combination product, out of 31 people, no hospitalizations, no emergency room visits, and a much higher rate in placebo. So this does seem to be the theme that emerges from a bunch of different studies, that you do have a good clinical benefit if you get this into people early. Next slide, please. Now, the other part of their development program for both products has, what about using these to actually prevent COVID in high-risk situations? So 2 situations have been looked at and with some information available. One is nursing home residents looking at symptomatic COVID, during an outbreak or a suspected outbreak, and you see about an 80% reduction in the proportion of people who developed symptomatic COVID who got BAM alone, kind of at a relatively high dose versus placebo. And also, interestingly, it's again a tidbit that, the numbers aren't huge, but you see no deaths in the BAM group and 4 deaths in the placebo group. So that's encouraging. Regeneron did the same idea, but in a little different way, where they looked at 400 patients in the same household as COVID-19 patients. And if you look at the development, so they gave the drug to people who were in a household where somebody had COVID. And what you see is 23 or 10% developed asymptomatic infection in the placebo versus 10 or half as many in the monoclonal antibody group. But they had a much lower viral load in the monoclonal antibody group. And maybe that would correlate with them being less infectious and less able to pass it on. And there were no symptomatic cases in the monoclonal antibody group, as opposed to 4% or 8% who developed symptomatic infection. So again, another signal that these may clear the lowest hurdle, which is to be used as a preventative. There is no EUA for that right now, so that's still a study application. Next slide, please. If you're going to administer these, though, you sort of need a plan. So they have to be given an IV infusion. Again, they are working actively on sub-Q, kind of like Hizentra types of ways to give it, which will make it a whole lot easier. But the uptake has not been what people thought. And I think it's in part because even though you don't have to buy these, the government buys them. You just have to apply and get a supply. But you've got to set up a system, and it's going to cost a bit of money because there is no difference between what you buy it for and what you're administered for. And there's still some billing, I think, for the administration, but it doesn't quite cover the cost. And I think that has discouraged some of the use. Next slide, please. We've been pretty aggressive here in my institution, where I think our administration did the right thing. And we actually hired, I think it was 1.5 pharmacists, and they just review all of our positive tests every day. And if it's an outside test, you can enter it into the system. They don't actually get the clinicians involved. If there's a positive test, they contact the patient, they see if they meet the EUA criteria, and if so, offer them treatment. And we did it that way so we could get it done really efficiently and get people in quick, and have treated many hundreds of people in this way. And these are the general EUA criteria. Outpatient, you can actually treat inpatients if they're admitted for non-COVID reasons. No supplemental oxygen if they're on baseline oxygen, it's stable. Mild to moderate disease, less than 10 days of symptoms, high risk of progression. Outside of that, you'd have to apply for an individual use, which generally Lilly does not support. Regeneron does in certain circumstances, but rarely in the inpatient setting. Next slide, please. I'm going to skip over this fairly quickly, but just say that even with these combination products, there is concern for escape mutants, and in some in vitro studies, you do see that some of the mutations that are out there, particularly with the Lilly product, will escape. I think Dr. Lauring, who's 1,000 times more expert in this, is a real authority on this. We'll talk more about it in a few minutes. Next slide. And finally, I just want to mention that there's a lot of call to kind of try and understand will these drugs help our severely immunocompromised patients who cannot clear COVID? Anyone, I think, in infectious disease in particular has seen these folks. This was one that we had in our center who just had, over 100 days of COVID, trouble clearing it, multiple doses of convalescent plasma, and whether or not there's a role for monoclonal antibodies, particularly if people have at least intact T cells, is unknown at this time. And there is concern that this is a great way to create mutants, is to continue to give either convalescent plasma or antibody preparation. So there is a potential downside to applying these as well. You can't do this under the EUA. If you were to do this, you'd have to get an individual use. And then the last slide. So in summary, monoclonal antibodies are really the only directed therapy with reasonable evidence base available to outpatients. I think as we accumulate more real world use, we do see that they work, but you've got to get people early. So what's really important to me is to let our patients know, particularly those who meet the criteria of being at risk, high risk for progression, which would be certainly most people with liver disease and certainly post-transplant, that if you are getting symptoms, go get tested. If you get tested, let us know so that we can get you monoclonal antibody early. A lot of things we don't know. What's the efficacy in people who have received vaccine? There is some concern in immunocompromised folks. So we've seen some recently about the ability to generate antibodies. So there still may be a benefit to this. We talked about the risk of variants escaping. And again, the role for people with kind of persistent, this isn't really long COVID, but persistent active infection. And with that, I'll finish, and thanks so much for your attention.
All right, so I'll just go ahead and start then. So my talk will be focused on the safety and efficacy of the messenger RNA and the viral vector vaccines, probably most relevant to a US audience, since these are the vaccine candidates that have led the way with emergencies authorization by the FDA and have been rolled out for use at large. Next slide. So my research program does have support from Pfizer and Sanofi, as we're involved in the COVID vaccine studies run by those two companies. Next slide. None of which would influence my presentation today. So we'll start with viral vector vaccines. So we do know that viral vector vaccines are a relatively newish approach. I mean, there had been a lot of work percolating, especially with some of the epidemic viruses, Ebola for one, and that it was first in 2019 that we had a vaccine approved for use in humans, which was a vesicular stomatitis virus vector vaccine for Ebola. Now, we know generally the construct for some of these that are viral vector, which can either be a human virus that's not expected to be pathogenic or modified to be non-replicating in the most part for the vehicles that we're using, or other non-human primate virus vectors. And the whole goal is to utilize the vectors as a vehicle to deliver the genome of interest. And in this case, it's the spike protein gene, which is incorporated into the viral vector and prepared and delivered. And obviously, this viral vectors simulate natural infection. And so they have certain advantages, like, for example, having the advantage of being able to stimulate T cell immunity and generally result in robust immunogenicity. As previously mentioned, the viral vectors are non-replicating, so they're not expected to be able to cause disease in the host, and they also do not alter DNA. So you have the Johnson & Johnson and AstraZeneca vaccines representative of that work. Next slide. And then the new kid on the block, which is the mRNA vaccines, like we're all aware of. This was, the technology and work that's percolated over decades and just really rose to prominence with regards to COVID-19 vaccination. Again, since we did know what the vaccine antigen should be from the history of the work done with prior SARS COVID virus, it was pretty clear that everyone wanted to target the spike protein. And so there's a lot of work that went into, designing mRNA construct that encode for the spike protein. There's a little bit of work that went on looking into what part, if we should have sequences that encode just for RBD, would that be sufficient, or the full-length spike protein? And that was elegantly done in some of the earlier phase trials. So, messenger RNA is delivered, the body produces spike protein, and that results in antibodies like we are aware of. A messenger RNA is fragile, and so it's packaged within a lipid nanoparticle construct to protect the RNA from degradation. And we know that messenger RNA technology has broad applications, including cancer treatment and for other non-COVID infectious diseases. Pfizer, BioNTech, and Moderna are the lead candidates. Next slide. So I just thought to show you the lead vaccine platforms. There's pros and cons of each of these vaccine approaches. The four that are widely used, not just in the US and globally, include the messenger RNA vaccines, Pfizer, Moderna, the non-replicating viral vector vaccine, AstraZeneca, that uses a chimpanzee adenovirus, and Johnson & Johnson, which is testing both single and double dose adenoviral to adenovirus 26 vector. There's inactivated virus vaccines, Sinovac through China, and then there are also Novavax and some other adjuvanted protein vaccines. Some of these are much more easy to produce than others. So, for example, the messenger RNA and the protein-based vaccines relatively. And some of them also have limitations, including we know that with the messenger RNA vaccines, the requirement for cold chain, Pfizer requiring more colder environments than Moderna. But since stability data have shown that maybe they don't need to be preserved at so much cold temperatures, and actually can be stable for much longer. The viral vector have an Achilles heel, which is anti-vector immunity, which I'll mention later, and also the inactivated viral vectors can be a little bit challenging to mass produce. But important for this audience, I think, when we think about immune compromised hosts, including people with advanced liver disease, we do know that inactivated viral vaccines are safe to use in those populations, as against some concerns about using live attenuated vaccines in immune compromised hosts. Let's go to the next slide. So I just thought to provoke you here with anti-vector immunity. We did mention that there's always the concern that pre-existing immunity to viral vectors can impact their efficacy, and we're going to learn so much more about that. And it's fascinating how different vaccine approaches has tried to evade this concern. So, for example, the Johnson & Johnson vaccine is using a higher viral payload in their vaccine, but also tested a single-dose vaccine so that there's no repeat exposure second dose afterwards. We saw with the AstraZeneca approach that a half dose followed by full dose against a full dose and a full dose appear to have even greater immunogenicity. And one of the postulates are about the some concern for anti-vector immunity. There's also been some data on increasing the duration between doses, again, like we saw with AstraZeneca, that appeared to be to have greater immunogenicity if you spaced out the doses. Using non-human primate adenoviral vector evades that, because so the chimpanzee adenovirus is one way to do that as well. And we saw with the Sputnik V vaccine that using two different kinds of adenoviral vectors, the Ad5 and Ad26 sequentially, was one way also to evade that. And in the UK, there's also vaccination combos that are being studied, including either receiving a messenger RNA vaccine ahead of a viral vector, or a viral vector followed by messenger RNA separated by 4 and 12 weeks. So we're going to learn quite soon about the immunogenicity and efficacy of different mix and match combinations, which is fascinating. Just thought to highlight that there's some geographic differences in adenovirus seropositivity for the human adenoviruses, where they tend to be much more higher seropositivity among Blacks and Asians compared to Caucasians in Europe and North America, which is important. And also just to think forward about viral vector vaccines, we know that if anti-vector immunity is a concern, then if we do need booster doses because of waning immunity or for new variants, they may be concerned about how anti-vector immunity should weigh into that. And also in populations that have been exposed to one kind of adenoviral vector for one disease, if the same vector is used as a vaccine vehicle for a different infectious disease, could the response to the second vaccine be mitigated by anti-vector immunity that has been developed with exposure to the first vaccine? So these are all very fascinating questions that we'll continue to learn more about. Next slide. So let's talk a little bit about vaccine efficacy. Next slide. So I just wanted to highlight some of these data. We knew how some of the preclinical data really provided very important information that helped us with the human phase vaccine trials. So this was a trial of the Ad26 vector. So obviously this was the J&J vaccine construct, and this was looking at it in rhesus macaques. So rhesus macaques were challenged both intratracheally and intranasally, so exposed in both upper and lower airways with SARS-CoV-2 6 weeks after they'd received different variations of Ad26 vaccine constructs. And so this allowed looking at both immunogenicity and also efficacy in protecting against upper and lower airway disease using different vaccine constructs, and provided important information. The bottom line here that I wanted to highlight is that this is where it became clear that antibody responses were developed typically between 2 to 4 weeks following receipt of the vaccines, and that also there was robust T-cell immunity generated by these. But that's where we had a proof of concept that these vaccines also can protect against carrying the virus in nasal passages and also establishing disease in lower airway. So this started to provide evidence about the possibility that the vaccines protect against clinical disease, but also could protect against viral carriage. I remember at the time when I reviewed this paper, I made a few comments in the bottom that I think eventually held true, that the single shot may work in humans and that the vaccine may not entirely prevent clinical disease, but if it occurs, it could be muted when contracted. And we see that that has held true with human trials. Next slide. So, we're not going to go through all the efficacy reports, but just want to make certain highlights here. So we do all know the efficacy data with the mRNA vaccines almost having identical or twin efficacy reports between Pfizer and Moderna. Both looked at all range of symptomatic disease from mild to severe disease. And then we also saw the efficacy data reported from J&J as 66% overall, a little higher in the US. But we saw that the numbers from South Africa, driven by the variant first identified in South Africa, resulting in decreased efficacy against moderate to severe disease as a composite outcome, but still having robust protection against the more severe forms of the disease. Similarly, for AstraZeneca, we saw a difference between the full and full versus the half and full dose. Again, most of you have heard that, again, in South Africa, the vaccine did not offer protection against the mild to moderate forms of the disease. And we see the Sputnik V vaccine looking at 2 different advectors showing that it had a 92% efficacy, again, against symptomatic COVID. So again, across the board, a little bit of differences in numbers, efficacy numbers clearly impacted by the variant identified in South Africa. But we'll look later on about the more severe forms of disease. The last point I just wanted to make here as well is that the trials are being expanded. Pfizer has completed studies in 12 to 15-year-olds. Moderna is conducting trials also within that same age group. Both platforms are going to study their vaccines in even younger ages as well. Same with Johnson & Johnson in the future. And studies for pregnant women, for example, have begun with the Pfizer vaccine starting the roughly third trimester of pregnancy. So it's great to see how the vaccines are progressing. Next slide. So one point I just wanted to make here is that even though there are variations in the degree of protection conferred against asymptomatic disease or milder forms of disease, on the more extreme spectrum of COVID-19 disease, which is severe disease, critical disease, and death, the vaccines across the board, both messenger RNA vaccines and viral vaccines have shown really excellent protection against severe disease and death, and in most cases, close to 100%. And in general, it appears that there seems to be a gradient that the vaccines tend to perform better and better with protecting against the most severe forms of the disease. But we do recognize that host characteristics are important, being elderly, having underlying comorbidities, or using certain medications that can mute immune responses may impact some of these numbers eventually. And also the presence and absence of circulating variants of concern may also impact that. But again, I think, thinking about, a common question we get, what vaccine should I get? Is one better than the other? I think the fact that they all protect against the worst forms of the disease and dying from the disease, regardless of which vaccine one receives, is really a remarkable achievement. Next slide. And one other point I wanted to make is, again, just this is the efficacy from the Johnson & Johnson vaccine, that really 2 weeks post-vaccine, that's where you start to see the separation within the trial for both placebo recipients compared to individuals who received the vaccine. And so that's why the term about when people are fully vaccinated is really defined as 2 weeks after you get the single dose J&J vaccine, or 2 weeks after you get the 2 series Pfizer and Moderna vaccines. We do know that with the messenger RNA vaccines, you already start to see protection occurring after the first dose, but being maximal after the boost, so after the prime boost approach. Next slide. So let's look at the vaccine trial participants. Again, for a liver disease specialty audience, to always ask yourselves, are the populations that were studying in clinical trial, do they look like the patients I care for in my routine clinical practice? So this was a characteristic of the phase 3 clinical trial populations. I pulled this from the FDA briefing documents, and you can see, this smattering of chronic diseases, hypertension, diabetes, obesity, but also, a minority of small numbers of individuals with liver disease. The study protocols were written in such a way that these were more likely to be those with mild liver disease, and usually individuals who had much more advanced liver disease or severe liver disease were excluded from these trials. Really important point to mention. And certainly, people who had received solid organ transplants also would have been excluded from these trials. All right, and so for many of these liver disease entrants into the study, some of their etiologies of their liver disease and severity were not always captured, but as you can see, the numbers are really too small to make much robust characterizations of their efficacy, or sometimes in those populations. Next slide. So let's start breaking down some of the subpopulations here. So across the board, regardless of the vaccine platform, whether it be messenger RNA or inactivated vaccines or viral vector vaccines, as you can see on the table on the right, if you compare younger people, typically to the left of the figure, to older people, typically to the right of the figure, you can see that the neutralizing antibody titers tend to be lower in older individuals. Okay, but if you look at the table on the right, next slide. If you look at the table on the right, you can clearly see that even though there's a little bit of a decrease in antibody neutralizing titers in older individuals, it still remains in some cases above the target range, which is the neutralizing titer levels in individuals, healthy people who've convalesced from COVID, which has been used as the bar. And we also know that in general, the level of neutralization titers don't always correlate to the degree of immunity against the disease. And I think we've seen that, at least from the studies that have reported their data so far, that older individuals appear to be enjoying, similar, or if not close to similar, protection against COVID-19 compared to younger individuals, in spite of these. Next slide. And then this is just data that is really contemporary. This was a research letter submitted to JAMA that I think most of us had seen. And this group asked a simple question: after receiving a first dose of Pfizer or Moderna messenger RNA vaccines, how did individuals who got solid organ transplants do? And so they checked roughly about the median time, I think, of 20 days after the first shot. they tested them for IgG to the spike protein, so looking for a vaccine response. And this wasn't quantitation. It was all about, did you have antibody detected or not? So just to be clear about that. And so they broke down the groups based on renal versus non-renal transplants, individuals who'd had transplants more than 6 years or less than 6 years, those who are on anti-metabolite immune suppression versus non-anti-metabolite messenger RNA, Moderna versus Pfizer vaccine. And overall, they found that, younger transplant recipients, those that received Moderna vaccine and those not on anti-metabolite immune suppression tended to have better responses. But also important to mention that across the board, regardless of some of these characteristics, that there was quite significant numbers of transplant recipients who did not develop antibodies to the first vaccine. And this really goes against, almost 100% in the clinical trials who developed IgG following the Pfizer Moderna vaccine. So this raises concern and really isn't surprising that immune suppressant medications can definitely modulate the response to vaccines, which is quite concerning. Next slide. So let's just probably end off with vaccine safety. Next slide. So I pulled the grade 3 reaction. So I know many of you may notice that the percentages here are not as high as you're aware of. I pulled off the mild to moderate and just left in the grade 3 side effects, and just really to compare those across the different vaccine platforms. And again, this is pulled from the FDA briefing documents for all 3 of the vaccines that have achieved emergency use authorization. Again, local reactions tend to be more pain from the injections, and typically, these reactions are worse following the second dose. And then systemic side effects, again, across the board, it's flu-like symptoms, fatigue, headache, myalgias. These tend to happen within the first few days of getting the vaccine and resolve pretty quickly within the same time frame. And, again, range quite across the board as shown. Now, important to mention, probably even more important, that when it comes to severe adverse events, you can see that those are very, very few. So non-fatal SAEs, as you can see, were very few across this clinical trials, but deaths related to the vaccine were did not occur, which is very, very reassuring. And we've also started to generate safety data in pregnancy. There's animal data both from Pfizer and Moderna about the vaccines not impacting fertility or resulting adverse reproductive outcomes, which has been really encouraging. Again, the safety data overall of these vaccines have really been stellar as they've been rolled out. Next slide. So we've all, I added this slide today because I know there's been a lot of buzz about, AstraZeneca being stopped from being rolled out in a handful of European nations in response to concern about what has been really very different, syndromes, including low platelets with hemorrhage in some cases, and in other cases, thrombotic events. So maybe very different pathogenic mechanisms for some of these occurrences. I just wanted to emphasize that as we roll out the vaccines and millions of people get them, I expect that there will be some of these false alarms around, certain background events coming to forefront. Again, many of these occurring at the background rate that they would occur in a population that were not vaccinated anyway, but getting attention because those individuals just received a vaccine. And just to put this in context, out of 17 million people that were vaccinated in the UK and Europe, it's really only been about less than 40 cases, I think about 37, in which these cases have occurred. So 0.000 per 100 patients, and not too different from the background rate, if not lower. I predict that later in the week, when the EMA sits on Thursday to review these, that they will definitely give a go-ahead to resuming vaccination with the AstraZeneca vaccine. Next slide. So in summary, again, we've seen very safe and highly effective vaccines, especially, you can literally guarantee anyone getting the vaccine that their risk of having severe critical disease and dying from COVID-19 would be unusual and unexpected. And I think that's just amazing to be able to say. Again, decreased immunogenicity in older adults didn't appear to be affecting efficacy, but we've seen this new data in solid organ transplants that actually, again, it was to the first dose. Those are prime boost strategies. So that was response to the prime, but at least start to raise concerns about certain subgroups that were not studied in clinical trials, including the patients that we mentioned having a diminished response and what that means. So I'll stop here, and I'm sure we'll have time for questions later.
Great. Well, thanks for joining in. I'll go ahead and get started. Next slide, please. So next slide. So these are my disclosures, mainly on antiviral drugs, so not particularly relevant to the current topic. Next slide. So today, obviously there's been a lot of talk about SARS-CoV-2 variants in the news. I just picked two from the New York Times, one here with Carl Zimmer talking about what is now known as the California variant. And then soon thereafter, this article from Gina Colada, also in the Times, which I had some good quotes. The term scariants was coined, as well as mutant porn. But I think the real challenge for the public was highlighted is, I think this Dr. Musser in Houston said, I think the crucial thing is all of this is that it's very hard for the medical community and lay public to really sort through all the noise about variants. At the end of the day, does any of this mean a hill of beans to anyone? So I'll take that as the challenge for my next few minutes with you to try to provide some clarity of which variants to worry about and which ones might be scariants. So next slide. So first, some terminology. A mutation is the actual change in the nucleic acid, the RNA, or the amino acid sequence in a viral protein. So ones that we'll talk about are N501Y, so that's asparagine tyrosine at position 501, or E484K. So that's the actual change in the sequence. But when we talk about variants, we're basically talking about two different sequences, two different genomes of the virus that are different. And so a variant may have multiple mutations, as we'll see. And then a lineage is sort of like a variant, at least these days, and it's kind of a variant and its descendants. So it's kind of like a subfamily of the SARS-CoV-2 family tree. And then a strain is technically a variant that is different in some way. It's maybe not as well targeted by the immune system, or it's more virulent. But I think today it's basically a garbage term because it really, it's used interchangeably. So next slide. So this is just a snapshot of, kind of the key variants that have been, being tracked lately and that have been in the news. And I'll just refer you, this is from outbreak.info, which is a regularly updated website that has nice graphics. You can explore these things. I turn to it almost daily when people ask me questions. So it's a good site to get into this on. The three main ones, which are variants of concern. So those are the ones that are problematic and everyone kind of agrees on. Those are B117, which is up here, P1, which is here, and B1351, which is here. And I'll spend most of my time talking about these. There are other ones you've heard about, which are variants under investigation, or VUI, or variants of interest, which means that maybe we need to be worried about them. Here's the California variants, B1.427 and 1.429, and then the quote New York variant, B1.526, which is shown at bottom right. So I'll just next slide, I'll kind of walk you through a few of them. The main thing, I mean, these variants have lots of mutations, but we pay most attention to the mutations in the spike protein. And that's because spike, of course, as you've just heard, is the antigen. It's what is targeted, it's in the vaccine, it's targeted by our neutralizing antibody responses, it's the protective response that we're looking for. And of course, spike also mediates how a virus binds its receptor and actually gets into cells and into us. And so that's why there's so much focus on mutations in spike, and that's where I'll mainly talk about, but that's not that these other mutations in these variants aren't important, but it's just, it's been secondary of late. Next slide. So lineage B117, which is known as the UK variant, I generally don't like geographic names for these things because of course they're everywhere and they're all of our variants now. But I do recognize that it's easier than remembering all the letters and numbers. So this was kind of the first one that was really hit global importance. It arose on what's called a long branch on the SARS-CoV-2 tree, which means that it had a lot of mutations when it was first discovered, more than you would expect. And this has led to speculation that it may have arisen at some point from an immunocompromised host because the virus evolves a little bit differently in those individuals. Importantly, it spreads way more rapidly, about 50% quicker than non-B117 viruses. That makes it more difficult to contain. That's why it's more transmissible. That doesn't appear to vary with age. There was early speculation that there were differences in how it behaved depending on kids versus adults, but that really hasn't held up to scrutiny. But in contrast, there is newer data does indicate, I wrote suggest that it may cause more severe disease, and there's been a battery of papers in Nature and elsewhere in the last week on these data. So, it does appear that there is a linkage to severity with this variant. Next slide. With B117, what I say, are we all Denmark? And this is basically showing what was, I think, first well shown and documented in Denmark, but now is been shown elsewhere, including in many areas in the US. So I have the US case counts from the New York Times, and then the percent of cases that are B117 in the other graph. And you can see that overall cases are going down. But then at the same time, we're seeing that more of our cases are B117. And I think this leads many to think that we are at this strange time period where we don't know yet if we can celebrate and whether things are going to be lasting. Because if you have a more contagious variant, that's what's left. That can lead to another wave of infection. So I think time will tell in the next few weeks or month. Next slide. B1351, also known as the South African variant, it has these mutations. It appears to also spread rapidly. There's concern that it's less neutralizable by sera, both post-vaccination and in people who've recovered from COVID. And it's thought that much of this is mediated by this E484K mutation, but also contributions from K417 and N501 as well. And that is different from what we've seen with B117. Next slide. P1 is known as the Brazilian variant. It also has E484K, like the South African one, and 417. So it's been shown to have explosive outbreaks in Manaus in Brazil, where there was a high amount of infection early on in the pandemic. So there's been a lot of reinfection, we think, and so that's been particularly concerning in terms of whether these people had pre-existing immunity and now this variant can still infect them. And so it has the same 3 key mutations, 417, 44, and 501 as B1.351, the South African variant. Next slide. So I'll spend my last couple minutes telling you about monoclonals and vaccines. So will monoclonals still work? Yes, but there are some issues. So E484K, as I've indicated, is particularly an issue for antibodies, and this shows it's bamlanivimab, I'll just call it BAM, which is the one antibody Lilly drug. E484K escapes that. There's also some discussion that L452R, which is prominent in the quote California variant that you might have heard about, also may escape BAM. And so that's really spurred combinations. So the Regeneron and also the Lilly combo. And so this graph just shows that with the Regeneron combo, there's really only a couple mutations that aren't too common. This E406W, can escape, but it's not one that's seen very much. But I think, with the Lilly, we don't quite know. It looks similar with the combo, but I think it just highlights that we need to watch not necessarily variants, but specific mutations for these monoclonals. But combinations should help. Next slide. So will the vaccines work? So this has been looked at in two ways. First, a couple over the last month or two, there have been a number of studies where people take sera from people who've received the vaccines and then tested them against the virus, in vitro. And this compares B117, P1, B1.351 in those first bars to a reference strain, USA Washington 1, which is one of the earliest strain in the US. And you can see that B117, the antibodies tend to neutralize it. P1, pretty similar. B1.351 there is a hit, several fold, in the ability of antibodies to neutralize that. And so that is, something that, people are watching and concerned about. I think the key here is, there probably is an effect, but this is a correlative protection. And really, if you want to know if a vaccine is going to work, the only way to know is if the vaccine actually works and protects you. So next slide. And so this will be my last slide. And so it's, what do the trials say so far? So there is some encouraging news, in my opinion. So the J&J vaccine, which you just heard about, in their FDA filing, that the vaccine was 72% in the US, 66% in Latin America, where P1 has been circulating, and 57% in South Africa, where 1351 was circulating at the time of the trial. So, that's reasonably encouraging that while there is a hit in protection, it's still largely effective. Novavax, which basically is press release data, also shows a hit with efficacy against B1.351, but preserved effectiveness against B117. And then Pfizer, we have observational data from the UK of people in the Siren cohort, which is healthcare workers. At a time when there was a lot of B117, 85% effectiveness 7 days after the second dose in this population. So encouraging that B117 for sure, appears to not evade vaccine immunity. And with that, I will hand things off to Dr. Rosso.
Thank you. And thank you to the moderators and the ASLD to invite me to speak today on therapeutic and vaccine recommendations in liver disease patients and transplant recipients. Next slide, please. So I'm going to be referring a lot of our statements or guidance to these two documents that are regularly updated and available at the ASLD website on the link on the bottom of the slide. Next slide, please. So as we move through the talk, we're going to move from patients with chronic liver disease with or without cirrhosis to transplant candidates, such as those with compensated cirrhosis enlisted with HCC and those with decompensated cirrhosis, and to the recipients or post-liver transplant patients. Next slide. So SARS-CoV-2 has direct and indirect effects on the liver. There may be direct viral cytopathic effects that cause liver injury. Liver injury may be secondary to the inflammatory or immune response. Keep in mind, we may see elevations in liver enzymes, particularly AST, due to muscle injury or myositis. And we're going to touch upon some of the hepatotoxicity that may be associated with some of the therapeutics for COVID-19, albeit rarely. Next slide. This slide demonstrates that the SARS-CoV-2 attaches to the ACE2 receptor, which has greater expression on cholangiocytes compared to hepatocytes. But despite this, clinically, we tend to see elevations more commonly in aminotransferases compared to the cholestatic enzymes. Mortality has been associated with alkaline phosphatase and AST, as well as lower levels of albumin. Next slide. On this slide, I am showing the prevalence of COVID-19 and chronic liver disease. And this is a meta-analysis of 73 studies that included over 24,000 patients. Among patients hospitalized for COVID-19, 3% had chronic liver disease, which is similar to the rate of those patients hospitalized for other indications other than COVID-19. Mortality was increased in the chronic liver disease 1.7-fold in those with COVID-19 and chronic liver disease. And whether or not chronic liver disease is prioritized for vaccination, for example, depends on the state. Risk factors for severe mortality or liver disease include alcoholic liver disease, HCC. NAFLD, the results have been mixed. Remember, NAFLD is associated with obesity and diabetes. If we could go back to the prior slide. NAFLD is associated with obesity and diabetes, which are associated with mortality from COVID. And cirrhosis has been associated with increased mortality, which I'll show you on the following slide. On this slide, we're showing data from the Secure Cirrhosis Registry demonstrating a stepwise increase in mortality as you move from Child-Pugh A cirrhosis to Child-Pugh C. And you can see the Child-Pugh C cirrhotics have a 9.3-fold increase in mortality compared to those without cirrhosis. The cause of death, however, in these patients, the most common cause of death is COVID lung disease, followed by liver disease. And at least in this report, alcoholic liver disease was the only etiology associated with mortality. The patients with cirrhosis were more likely to be treated with the agents that we don't use anymore. You can see on the left hand of the slide compared to those without cirrhosis. Next slide, please. So the guidance document recommends the following for the treatment of underlying liver disease. So let's look at individuals with chronic liver disease who have COVID-19. You can see here that hepatitis B therapy can be initiated, and keep in mind that some of the therapies that we use for COVID-19 have immunosuppressive properties and may provoke reactivation of hepatitis B. Otherwise, for hepatitis C and PBC, therapy could be deferred in the setting of COVID-19 disease. For patients with autoimmune hepatitis, consider reducing or discontinuing immunosuppression. However, there may be exceptions, such as those with severe autoimmune hepatitis, where therapy may be indicated. And those who are undergoing surveillance for hepatocellular carcinoma and have COVID-19 disease, their surveillance can be deferred until infection with COVID-19 is resolved. As we move to the right side of the slide, you can see essentially for all chronic liver diseases, initiate or continue therapy in individuals who do not have COVID-19, as well as continue surveillance for HCC. Next slide, please. So the management of hospitalized patients with chronic liver disease and COVID-19 really should be the same as those without chronic liver disease. We'll discuss a little bit how tocilizumab, the prescribing information states to discontinue if the ALT or AST are more than 5 times the upper limit of normal. For remdesivir, the recommendation, the prescribing information is to consider discontinuing if ALT is greater than 10 times the upper limit of normal. Patients who are hepatitis B surface antigen positive treated with dexamethasone or tocilizumab should be on antiviral therapy for hepatitis B. Remember, I had mentioned about the potential to provoke reactivation. Consider reducing or discontinuing drugs associated with leukopenia or lymphopenia, such as the antimetabolites, azathioprine or MMF. AST and ALT elevations more than 5 times the upper limit of normal may exclude patients from clinical trials. And tacrolimus should not be routinely reduced or discontinued, which I'm going to show you the reason why in the following slide. In this study that included 36 centers from 9 countries with 243 liver transplant recipients with COVID-19 from the European Liver Transplant Registry, patients with COVID-19 were observed either at home, on the hospital ward, or in the ICU. There was no mortality in those who were followed at home. Mortality was 17% for those on the ward and increased to 54% in those in the ICU. In this study, in multivariable analysis, tacrolimus was independently associated with a 45% reduction in mortality. Age greater than 70 was associated with a 4-fold increase in mortality compared to those less than 60. The authors speculated that perhaps tacrolimus blunted the immune response that is seen, or that cytokine storm that is seen with COVID-19 as one reason why tacrolimus was associated with a reduction in mortality. I think there'll be more to follow on this. Next slide. On this slide, I'm showing some of the more common therapeutics used to treat COVID-19 disease. And as you sort of scan across and look at the metabolism and the regimen used, if you get the dose adjustment column, you see no dose adjustment is recommended for those with chronic liver disease. And liver test elevations are either rare or relatively uncommon. such as remdesivir, grade 4 elevations to 2% of patients. And we discussed the recommendation for discontinuation with ALT elevations more than 10 times the upper limit of normal. And with tocilizumab, 2.9% had ALT elevations greater than 5 times the upper limit of normal. And the recommendations for discontinuation are shown there. Next slide. So should patients with stable chronic liver disease receive the COVID-19 vaccine? First, I hope you'll note that look how many patients were included in these randomized trials. For Pfizer-BioNTech, it was 16 years of age or older were included in the trial, with more than 21,000 in the vaccine group, of which 124 had chronic liver disease, most of which had either chronic hepatitis B or C. However, the efficacy and safety were not reported in this subgroup yet, a relatively small subgroup. In the Moderna trial, which included individuals 18 years of age or older, more than 15,000 were in the vaccine group, 100 had stable chronic liver disease, and in the briefing document, no patient with chronic liver disease in the vaccine group developed severe COVID-19. Next slide, please. Anaphylaxis to mRNA vaccines is reported through the Vaccine Adverse Event Reporting System, and from over 1.6 million vaccine recipients from December 14th, 2020 through January 13th, 2021, relatively equally split between the Pfizer, BioNTech, and Moderna vaccine. You could see that anaphylaxis was reported in 4.5 cases per million. And this is not too dissimilar to vaccines that we already use, like influenza, pneumococcal, and zoster vaccine. Almost all of the patients who had developed anaphylaxis were women, and almost all of these cases occurred within 15 minutes. Next slide, please. For the Johnson & Johnson vaccine, again, 18 years of age or older, of which more than 21,000 were in the vaccine group, 103 had chronic liver disease. There were 7 immunocompromised patients from solid organ transplant included in this. And in the FDA briefing document, it said that 1 patient with chronic liver disease developed moderate to severe COVID-19 disease 14 days after vaccination. There were no cases of anaphylaxis immediately reported following vaccination, and there were 5 cases of non-serious urticaria in the vaccine group and 1 in the placebo group. Next slide, please. So should patients with chronic liver disease receive the COVID-19 vaccine? Yes. Do not withhold their medications for chronic for their liver disease. Patients with fever or current infection should wait until stable. Administer the vaccine at recommended intervals and not more than 6 weeks apart. Do not restart the series if longer than a 6 weeks delay, and administer at least 14 days apart from other vaccines. Wait a minimum of 90 days to vaccinate after the COVID-19 infection if the patient received monoclonal antibody. Next slide, please. Recommendations for COVID vaccination in liver transplant candidates and living donors. Liver transplant candidates should receive COVID-19 vaccine prior to transplant whenever possible. Healthy living donors should receive the COVID-19 vaccine electively and preferably prior to donation. But even after vaccination, we must remind our patients that COVID-19 precautions, including masking, hand washing, social distancing in large groups, should be adhered to. Next slide, please. So what about COVID-19 vaccine in solid organ transplant recipients? This was mentioned a little bit earlier. This is a study in PRES and transplantation. 187 solid organ transplant recipients were included in this group, of which 35 had liver transplant. Most were on tacrolimus-based immunosuppression, but almost 3/4 were on antimetabolite. Again, equally split between the Pfizer, BioNTech, and Moderna vaccine. In this group, there were no self-reported SARS-CoV-2 cases. There was no rejection, no neurologic complications, and no allergic reactions that required epinephrine. There was a case of the flu and a case of pouchitis. And then the most common symptoms tended to be constitutional symptoms listed on the bottom on the right side of the slide. Next slide, please. Patients who developed antibodies after a single injection were reported. It was 31% in the Pfizer-BioNTech vaccine and 69% in the Moderna vaccine. I think that before we say, is Moderna better or not, we need to wait for the final results from the study. This is a preliminary analysis, and I think sort of there's sometimes a haste to get the information out there, and I think we need to just be careful as we look at this data that we need to wait for the final results after the second injection is given before we reach any conclusions. Next slide. So in the next few slides, we'll conclude. So COVID-19 vaccine is recommended for patients with liver disease, with all etiologies of liver disease, whether they have compensated or decompensated cirrhosis or hepatocellular carcinoma and post-liver transplant. Contraindication to the vaccine includes a history of an immediate allergic reaction to PEG or polysorbate. A history of Guillain-Barré or food allergies are not contraindications. Next slide. So as we walk across the timeline here, so patients with chronic liver disease with or without cirrhosis vaccinate and continue medications. Transplant candidates, whether compensated or decompensated, vaccinate listed patients, vaccinate living donors. If transplant interrupts the sequence, try to give the vaccine around 6 weeks, 4 to 6 weeks after transplant, so that there's less intense immunosuppression, or certainly our post-transplant candidates should be immunized following liver transplantation 6 weeks later when there's less intense immunosuppression. Thank you, and that ends my presentation.
Great. Thanks to all of our presenters for really outstanding presentations. All of these presentations will be available through the AASLD website if you've missed part of them or want to go back and review the copious amounts of information that have been presented. Our panelists have been very generous with their time and are willing to stay on for just a little bit longer. And so we thought we'd just sneak in a few questions before concluding this webinar. I think one of the first things that we're really interested in is really what the impact of yesterday's publication in JAMA has on the transplant population. So I'm going to start with asking Dr. Ogbuagu about whether or not he thinks we should alter our practices or make any recommendations to our transplant patients based on the JAMA publication that you mentioned in your study.
Yeah. Okay. So here are a few thoughts. We should not be surprised that drugs that modulate B and T cell responses would diminish responses to a vaccine, right? It makes total sense. So I think, that's pretty clear. Second of all, that the vaccines that were studied are 2-dose vaccines, and this was what happened after the first dose. We do know that the boost received from the second dose does result in much more robust antibody responses. So we still don't know if you could salvage, if you will, some of those cases with a booster dose of vaccine. But again, I think the fact that only 17% had positive antibody testing, it's very concerning and makes you wonder if in certain subgroups of patients that we need to start to think about ways to assess if they had appropriate responses. Again, remember that antibodies that are measured are not the full immune response to vaccines. There's T cell responses and other things that were not measured in that cohort. And I think the last presenter showed data from transplant recipients that none had breakthrough COVID, right? So that it appears that, again, we have to see if, again, this either delayed or diminished immune response would translate to, vulnerability to having a clinical disease. So I think, let's put to be continued on this one. I think we'll learn, so much more. I'm sure that cohort will probably also be looking at the same qualitative IgG presence after the second dose. So I think we'll learn much more even from that one cohort. One last point to make would be, should we then think about higher dose vaccines? For example, I participated in the Novavax phase 2 study, and we used escalating doses of the vaccine, and we actually included people who had received transplants but were at least 6 months out, and people who were 90 days out from chemotherapy. So I think as time evolves, we start to also learn more about how different categories of immune suppressed patients respond to vaccine.
Great, thank you.
Thanks so much, everybody, for all your talks and for sticking around. Dan, this question's for you. We've had a lot of questions about the CDC's recommendation for waiting 90 days after getting monoclonal antibodies before getting vaccinated. Well, why is that recommendation there? And does that also apply to people who've had COVID but not gotten the monoclonal antibodies?
Yeah, thanks, Lauren. So for the question about monoclonal antibody and waiting, I think there's concern about two things. One is the antibody hangs around for a while, so you may be protected for a little while, but more importantly, it may bind to antigen as it's made and may reduce the immune response that develops. And so it does have a relatively long half-life, these monoclonal antibodies. So I think they sort of doubled it and said maybe wait 90 days. In the second question, regarding previous COVID, so that's a really interesting one. And, there aren't formal recommendations to limit the number of doses. I think anyone who is in infectious disease gets lots of calls from either colleagues or colleagues' patients or your own patients where they really have a very vigorous response to the first vaccine, sometimes with lymphadenopathy and much more significant pain than most people have. And then there's all this data that looks at antibody levels where if you've had COVID, you get one shot, you don't really look that different. You actually, I think, look a little bit higher if I recall. Yeah, I got it right. Thank you. Then people who, when you're not sure, it's nice to get some reinforcement that the antibody levels do look pretty good. And so it's, I think, very reasonable, especially if someone had a significant adverse event from the first one, a local type of adverse event, to hold the second dose. But nonetheless, I think for most people and immunosuppressed people, especially people maybe who had mild disease and may not have generated as much a response, it's reasonable to go ahead and get vaccinated, especially as supply increases.
Thank you. Adam, I wonder if you could comment on if we were really to have a choice, a full menu of vaccines, whether or not you think we should consider variants as we address which vaccine our patients should be getting.
Yeah, I mean, it's really interesting. I think, unfortunately, my conclusion is the data just isn't good enough to tell us that. And I think, as others have highlighted, I suspect that they're not as far apart as we think, and having to do with when the trials were done and where they were done. So I'm not sure we can say that one's going to be better than the other, in terms of the variants. I mean, I think it would, and it's also going to be, lumping all the variants together is challenging too. I think the promising thing in thinking about them is, that the mRNA vaccines, and to a smaller extent, lesser extent, the vector vaccines are very, not easy, but much easier to update than other vaccines. And of course, many will know that there's an ongoing study of a mixed B1351 and kind of standard SARS-CoV-2 Moderna preparation that's ongoing. So I think we'll know more over the next 6 months how this is going to change the vaccine landscape for sure.
Could I also ask Adam, this is Bob Fontana here.
Yeah.
The viral variants, they make it more to the newspaper than to the doctors. So how do we even know if there's viral variants in our patient population? I mean, tell us a little bit about the testing for that. I mean, is that even practical to be thinking about this?
Yeah, well, at our center, we're doing it, because this is what I do. This is my day job. So I think, but yeah, is it practical? Not really. In that, so the testing, there's the way we know most variants is that they're, is by sequencing, which, takes time. Even the best we've ever gotten on a turnaround is like a week from sample collection to sequencing. So that limits, its utility for, deciding whether someone should get a monoclonal or a convalescent plasma, et cetera. And so there are, assays being developed. I don't think any have like an EUA. There's certain, LDTs popping up for, variant specific, RT-PCR tests, that have primers and probes that differentially. One is the TAC path assay, which is commonly used. It loses one of its, its 3 targets, and one of them turns negative with the B117. So I expect we'll see more of those, and that might be useful. But sequencing will still have a role, and the turnaround's just not, going to be fast enough. I mean, you look, with HIV, where this is important, and we get an HIV genotype, it takes some time to get the data back. So I think, but I think where it is useful, where we found it useful at the University of Michigan is aggregate data over time where, we can track how some of these mutations are in our community, which we can then use to decide, whether to emphasize, the cocktails versus, BAM alone based on what's out there in our patient population. So that might be something we'll see more in the future.
So
Thanks.
more at the population health level rather than the individual patient level.
That's what I think, but both the time and the regulatory elements in terms of actually using it for clinical decisions, I think would be a big hurdle.
Thank you. Mark, this question's for you. We're hearing some data that the antimetabolites may be harmful in terms of patients that get COVID or for the response to vaccine. Tacrolimus may be beneficial. Should we be doing anything with our immunosuppression before a patient gets COVID, maybe in preparation for a vaccine? And what are you doing with your patients that actually do come down with COVID, even the mild to moderate forms?
Yeah, good question. This is sort of a very practical question. For the vaccine, we haven't changed, we haven't modified our immunosuppression for the vaccine. So to answer that, we leave the patients on their baseline immunosuppression. For the hospitalized patients, we have reduced the antimetabolite. I'd be curious to see what other centers are doing, but there have been associations with leukopenia, lymphopenia, and mortality. So if patients are sick enough to be hospitalized, we have either reduced their MMF, they're usually on MMF, not azathioprine, or even stopped it depending on how sick they are.
Great.
Thank you.
And not making any changes in tacrolimus, it sounds like.
No, it's we haven't made it even prior to that publication. We haven't really made major changes to the tacrolimus. I mean, we may, if they're sick and hospitalized, we may have lowered their dose a little, tweaked their level a little bit, but not. I wouldn't say that was a routine thing done. It was more with the antimetabolite.
And Dan, do you think there's a role for reducing the antimetabolite just to help get our transplant recipients to respond better to vaccines?
I mean, I wouldn't do it right now. I think we need more analysis. I mean, when do you stop it? How long do you stop it? Do you put it back on after the second dose? I mean, I wouldn't want to do that on one letter to the editor. I'd be interested actually what Emily thinks as well.
Yeah. I mean, I think that we have to be really careful about over-interpreting the results from yesterday's letter to the editor. It's interesting data, it's provocative data, but remember that when we looked at mRNA vaccines, the seminal trials of them really were looking at 2 doses. And I think Onyema said this, and I'm going to just reemphasize, let's wait, let's see the results after with larger numbers and after 2 doses, and then go from there. I think every time we make a decision based on very limited data, we walk it back a few weeks or months later. And so it's important to actually pay a little more attention to this. I do have one more question for Onyema, which has to do with the CDC guidance about not giving COVID vaccine within 2 weeks of another vaccine, and whether you think that makes sense and if it matters for some vaccines but not for others.
Yeah, honestly, I'm not sure where that's coming from. I can imagine when there's new vaccines and there's concern for maybe safety events, or at least just the need to monitor and catalog those, you want to separate them so you can distinguish what's one reaction to another. We vaccinate people with multiple vaccines all the time. Measles, mumps, rubella, TD, and all those things, right? We give more than one vaccine antigen all the time without jeopardizing response to either one. There's some vaccine combinations or sequence of vaccine combinations, for example, how you sequence PCV and the polysaccharide, which one you give first does matter and what the timing in between doses are. So in certain described cases, but that tends to be the exception, not the rule. So I really don't see any significant rationale for why someone can get the flu vaccine and COVID vaccine together, frankly, for now. So I think that, that's really a soft recommendation.
Okay, thank you. Well, it looks like we've exceeded our time by quite a bit. So I think we're going to wrap this up now. I want to thank the presenters again for really outstanding and thought-provoking talks, and to AASLD for sponsoring this, and especially to John Lingerfeld, who has helped organize all of this. I want to remind you that you can go on the AASLD website to review these talks and listen to salient points again. So thank you very much to the presenters and to the audience for sticking with it through past the time. Thank you.
Thank you, everybody.
Bye-bye.
Take care.
Bye.
Thank you.